Hello and welcome to part 3 of this three-part series on introductory microbiological topics. Again, this discussion, this lecture is designed specifically for my micro college students, but of course anybody can watch this and hopefully learn something from this lecture. So again, this is part 3. This is a final part of our first chapter of this microbiology course. So we'll look at microbes and human welfare. So microbial ecology. So this is where we go out into the environment and we study the relationships between microbes and their environment, with humans, with other microbes, with animals, with plants, and so forth. So we look at bacteria specifically and how do they function within the cycles that are out there, the carbon, nitrogen, sulfur, and phosphorus cycles. They are key. They are instrumental to all 4 of those cycles in the breaking down of compounds and releasing these elements to then be taken up by plants and then can be taken up by animals then after that. So they're the basis of the food chain. They're breaking down these compounds that are needed by all living things. And so microbial ecology is the branch of microbiology that studies these relationships. Bioremediation. So this is taking these organisms, living organisms, that's the bio part, and we're going to use them to remediate or to fix an environment. So for example, bacteria degrade organic matter in sewage. They can be used to detoxify pollutants such as oil and mercury. So as you can see, microbes are not always the bad guys. We use them in so many beneficial ways, and so we can use them ourselves. The picture that you see here, these 2 guys, this is on Valdez Sound. There was an oil rig that had run aground in Valdez Sound off the coast of Alaska, and it ripped a huge gash in the hull of this oil tanker, and millions of barrels of oil were spilled. And so we used bacteria on a large scale to break down the crude oil that had come out of this oil tanker. That's bioremediation, and we're using it for the Great Pacific Garbage Patch that's out there, the big plastosphere or whatever you want to call it, that's in the Pacific Ocean between California and Hawaii. That's just, there's a couple of them that are floating around. There are others around the world as well, but those are the most popular ones. And we're trying to genetically engineer bacteria to eat plastic and get rid of these giant floating islands of mainly plastic garbage that's floating around. That's bioremediation. We can use bacteria as biological insecticides. So rather than just crop dusting and spraying out these insecticides that kill everything, they're totally non-selective, very much like antibiotics. Antibiotics are non-selective. They kill, if it's a bacterial cell, they kill it. These synthetic insecticides can be used, and they're very effective in killing the pests, but they also kill the beneficial insects as well. So how do we save the beneficial but kill the pests? Well, we found there's this humble bacterium called Bacillus thuringiensis. We've genetically modified and engineered it as well, a lot too, through agricultural research and so forth, to get it to kill specific pests. So we put the bacteria all over the crop. These bacteria are harmless to animals and plants, but they are fatal to the pests that live on and munch on our crops. So with the bacteria on the leaves of the plants, the larvae, which is what these pests are, that are constantly eating, voraciously eating, they're eating the leaves, but at the same time they're eating the bacteria. The bacteria then grow within these, call them caterpillars, I suppose. They grow within the caterpillars and they basically just destroy their insides. And so they never can get, and what's most important is not only is it killing that organism, but it prevents the larvae from getting to the adult stage, which is the reproductive stage. So if we can stop these pests at their larval stage, they can never produce eggs and they can never make the next generation. And that's what these bacteria do, and they do it very well. So these guys are selective. They're only getting the ones that are actually eating the plants, not the pollinators. The pollinators aren't interested in that. This bacteria, they're killing the pests that are causing millions of dollars of crop damage. Biotechnology, the use of microbes to produce foods and chemicals. So we're using technologies to modify these organisms. So genetic engineering is an example of one, relatively new technique for biotechnology. Bacteria and fungi can produce proteins, including vaccines and enzymes and things that are needed by humans in order as therapies, for example, as therapeutic agents. We're genetically modifying them. Genetically modified bacteria used to protect crops from insects and freezing so that the crops can withstand the lower temperatures. So this is all new ways that we are taking bacteria, messing with their genes, genetically modifying them in order to benefit humankind. And it's working out really well. There's a lot of opposition to it, though, because when you genetically modify it, what's happening? Because when you genetically modify something, they're going to produce new proteins, and we're not sure how, say, humans, when they consume these proteins, how are their bodies going to respond to it? There's lots and lots of research out there. Just about everything we eat in this country has been genetically modified in some way if you buy it from the grocery store, unless you're totally gardening at home. Even then, you're using seeds that have been genetically modified in some way. We'll talk a lot more about this particular topic later. Microbes that are normally present in and on the human body are called normal microbiota. The older archaic term was normal flora, and we called them flora because we weren't sure where to classify them. They weren't animals, so we thought bacteria were plants, and so we called them flora. But I suppose the more sophisticated term is microbiota. Normal microbiota, living on our skin, on the conjunctiva of our eyes, living in our large intestine, and so forth. They prevent the growth of pathogens. So are they of benefit to us? Well, pathogens would normally want to colonize us. Well, the normal microbiota prevent pathogens from colonizing because our bacteria that are living on us and in us want us. They want to reside on our skin. They want to live in our large intestine, and they're going to protect their turf. We call that microbial antagonism. They also produce things for us like folic acid and vitamin K. We'll talk a lot more about that later. Resistance is the ability of the body to ward off disease, and so we'll discuss ways in which we resist pathogens, like through our skin, stomach acid, mucous membranes, cilia, antimicrobial compounds, complement proteins, and so forth. These are all different ways that we can ward off disease just innately, naturally within us. Normal microbiota, they assist in that resistance and building up resistance, which we'll talk about in another chapter. Some infectious diseases. Well, before we talk about a disease, what is a disease? When a pathogen overcomes the host resistance, disease results. When homeostasis within an organism has been upset, knocked out of balance, that's disease. And so I just wanted to conclude this chapter with discussing a few what are called emerging infectious diseases. These are diseases that are being watched by the WHO, which of course we hear a lot about these days, and the CDC as well. So what's causing the increase of these EIDs, emerging infectious diseases? Evolutionary changes in the microbes, spreading of diseases to new locations through increased international travel, ecological changes. There's political unrest will do this as well whenever a country breaks down and the government collapses. People are not getting health care, and so diseases break out, and these organisms can mutate and become new types of diseases. We're dealing with that right now with COVID. This is one that has mutated to a point that we did not expect, and it's a coronavirus, and coronaviruses we have been dealing with for centuries, if not millennia. But now with this one, with COVID-19, we're dealing with a newly mutated one that causes all sorts of problems within us. That's an emerging infectious disease. So I guess you could say COVID has emerged. It's become a pandemic and it has spread around the world in a very, very short period of time. And I want to emphasize that. As far as we know, that COVID emerged from Wuhan in late 2019, November, December, and here we are just a few months later. It's spread so rapidly, which is unusual for microbes and pathogens to be able to do that. Normally it takes years for this to occur, to spread at the scale that COVID did. This only took a few months. So these are ones that you've heard of. I'll go through them quickly. It's just a few emerging infectious diseases that are being watched now by WHO. West Nile encephalitis, or what's called the West Nile virus, first diagnosed in the West Nile region of Uganda in 1937. So West Nile has been around for a long time. It appeared in the United States in 1999 in New York City. How does it spread? Mosquito bites an infected bird carrying the West Nile virus. The mosquito then can transmit that virus after biting the infected bird to a human or some other host. And this is how the virus disseminates itself into a community through the bite of the mosquito. Bovine spongiform encephalopathy. This is mad cow disease caused by what's called a prion. We'll talk about prions later. So cows contracted the disease by eating infected sheep parts. And then humans were infected when they ate the infected beef, so it was passed on. And this picture is a photograph or video shot from in the mid-90s whenever mad cow disease broke out in Great Britain. They killed a couple of million, at least 2 million cows they exterminated because they wanted to stop the spread of this disease called mad cow disease. And mad doesn't mean like I'm so mad, I'm angry. No, mad in Great Britain means you're nuts, you're crazy, you're insane. And that's the way the cows kind of presented themselves. They were acting like they were nuts, like they couldn't think straight. They were falling down, and what was happening was their brains were being degraded by this infectious protein called a prion. E. coli 057H7. So this is not the E. coli that's living in our large intestine. This is a toxin-producing strain. It produces a toxin called the Shiga toxin, which creates real problems in the lower GI. It's the leading cause of diarrhea worldwide, and it was first seen in 1982. Recent outbreaks in the U.S. have prompted public health officials new methods for testing foods to see if the foods contain E. coli 057H7. Invasive group A strep, also known as the pleasant nickname of flesh-eating bacteria, rapidly growing bacteria that cause extensive tissue damage, increased incidence since 1995. It's also called just GAS, G-A-S, for group A strep. This is not the same strep that causes strep throat or scarlet fever. This is a different type of strep. It's a very virulent type of strep, and it spreads very rapidly and causes necrotic tissue wherever it's growing. Usually the only way to stop it is surgical debridement or amputation. Ebola hemorrhagic fever, which you've heard of. We're going through another outbreak right now at this particular time in June of 2019. But of course, it's not a big breakout. It's somewhat minor, but of course that's being eclipsed by COVID and our concerns with that. But Ebola has had a number of outbreaks over the past few decades. And it's caused by a virus. It's a hemorrhagic fever, so it's causing fever and hemorrhaging and blood clotting, first identified near the Ebola River of the Congo. Every few years there's an outbreak, and it spreads pretty rapidly as well, and has pretty high mortality rate. Hantavirus, I remember this from 1995. The media had covered it somewhat, and there was some scare. First identified in 1951 in Korea as a cause of hemorrhagic fever named for the Hatan River, and first seen in the United States in 95. So what happens is it comes during rainy seasons, so more, if you look in the little graphic there, more rain equals more food equals more mice equals more poop, and then the poop dries out, and this guy here you can see is showing an example. You're sweeping it up. What happens is then the dried fecal matter becomes airborne, carrying the viruses with it. You then breathe it in, and then you develop hantavirus pulmonary syndrome. So it's a respiratory symptoms. AIDS, of course, you've heard about. It's a disease caused by HIV, human immunodeficiency virus. In 1981, first time it was identified simultaneously in San Francisco and New York City, pretty much at the same time. Physicians didn't know about it at the time that it was in both cities, but then whenever the news about this disease was starting to be put out into medical publications, then doctors realized we're dealing with a new disease here. Worldwide epidemic infecting about 40 million people every day. We have 14,000 new infections, so it spreads very rapidly. Sexually transmitted disease affecting males and females. So we're still dealing with this. Most of the infections are in sub-Saharan Africa. About 70% of those 40 million infected people are in sub-Saharan Africa. So it's a real problem in that continent. Avian flu, called the bird flu, it's H5N1, caught public attention in 2003 when it killed millions of poultry and 24 people in Southeast Asia. So we need to watch these ones. The flu mutates, and so you don't hear about it, obviously, but it's one that's being carefully monitored still. There are so many diseases that are being monitored and watched by the WHO and the CDC that we have no idea that they're doing, because they don't want an outbreak to occur. So this is one that's still being watched, even though it was 17 years ago. It's still being watched closely. Normally, subtypes of influenza A do not spread from species to species, but this one did, and that's the scary part about the flu, is its ability to mutate. It's called antigenic shift and antigenic drift. And so this has to do with the DNA mutating, which we'll talk about when we discuss viruses later. Also in around 2002-2003, along with the bird flu, there was SARS. Would be called SARS-CoV-1, which it wasn't called initially because we didn't have a second coronavirus. This is the first coronavirus, SARS-CoV-1. It quickly caught health officials' attention because it spread worldwide in just a year, caused by a new type of resistant coronavirus and has no cure. Now, this is nothing compared to CoV-2, which is, that's the official designation of the one that we're dealing with now, SARS-CoV-2. This one killed just a little over 1,000 people globally. It was stopped, and it didn't spread as rapidly. Its transmission rate was much less than COVID-19. Its ability to be transmitted was far less than this one. I'm sorry, this one, it did not spread nearly as rapidly and as quickly as SARS-CoV-2. So it's in the coronavirus family, same one that we're dealing with now. And it spread pretty rapidly, and it was just started by one guy, a doctor who stayed in a hotel in Hong Kong. Cryptosporidiosis. This is caused by a protozoan called Cryptosporidium. Gained much attention in 1993 when over 400,000 people developed diarrhea in Milwaukee, Wisconsin. That really raised a stink. Sorry. So a lot of people developed diarrhea. What happened was crypto got into the water supply, the public water supply, and then it got into homes, and people drank the water, and crypto it exploded in their reservoirs and in their public water supply. Modes of transmission: contaminated water, like I said, through drinking water, but also in swimming pools as well. What happens is people release the eggs from in their feces, and then it gets in swimming pools, for example, and then floats around in the water. People then will consume the water through their mouths. They consume the eggs, and now they have crypto. That's the way polio was disseminated. It was a fecal oral route, is what it's called. So the infectious agent gets released in the feces and then gets consumed through the mouth. Also, contaminated hospital equipment can transmit it as well. It's a pretty bad case of it. I never had it. My father-in-law had it, and he suffered pretty severely. He didn't need to be hospitalized, but it lasted. He had diarrhea for about 2 weeks, and it was very unpleasant for him. Couldn't go to work, had to stay home. But he developed it from working at a job site using a porta potty. And the CDC came to their house because it's an emerging infectious disease. So when this gets reported, CDC is alerted. CDC sent a couple of people out to my father-in-law's house, wanted to interview him and try to figure out where he would have picked it up from. Well, that ends Chapter 1. This is the introductory chapter to microbiology. This is part 3 of a 3-part lecture series on Chapter 1. Hope you enjoyed this discussion, and thank you for watching.